Continuing here our discussion on B cells. So just to recap this real quick, once the body has been exposed to an antigen and has triggered the formation of an antibody, we will have what's called memory cells. And these memory cells can be easily and quickly converted to plasma cells that then actively secrete antibodies. So the principle of vaccination is that the body's exposed to the antigen without the need to get sick, and it's making the correct antibody. It will keep some memory cells around, and once the real pathogen hits, then these memory cells will be quickly converted into plasma cells, and the secondary immune response is immediate, and the antibody concentration is high right away, and the person will not get sick. So here, nice and big and large, hopefully convincing those of you that might be on the fence about vaccination, how great vaccination is. You will have the memory cells that you need, that you can quickly convert into plasma cells, and the plasma cells will churn out antibody very quickly, and then a secondary immune response is right away. It's immediate, and the body won't even get sick. So that's why everybody's looking for for a vaccine to the coronavirus. Okay, so here sort of the text right up to what I just mentioned, the switching back and forth between plasma cells and memory cells. Plasma cells, remember, those are cells that are actively secreting antibody. And memory cells are a small sample of these cells that have learned to make a specific antibody. They stay back, they stay dormant, and if they're needed, they can be converted quickly back into plasma cells that then actively make the antibody. So basically that's what it says right here. So the once a plasma cell has churned out the antibodies that were necessary to heal the body, then a few memory cells remain behind and waiting to respond to the same antigen, and that a lot of times this ability to respond to the same antigen quickly, that will be retained for a lifetime. So let's take a look at what antibodies are. They are proteins secreted by plasma cells. The typical, what we would normally think is the typical antibody is the IgG. So there are 5 classes of antibodies. They're also called immunoglobulins, and that's where the Ig comes from, the immunoglobulin, and then here would be the type G immunoglobulin. That's the one that's produced by these plasma cells in a secondary response, and it's the typical letter Y-shaped molecule that we'll take a look at in a sec. IgAs, they basically look like 2 antibodies connected to one another, 2 of those letter Y-shaped ones. So I can draw this maybe right here. So it's the IgA kind of looks like this. It has sort of this letter Y structure, but then a little bridge to another second Y-shaped molecule. So that would be more like an IgA. The IgE and IgD we don't really need, but the IgM, that one, is associated with primary responses. It looks more like a snowflake. It's like as if you're putting 5 of these IgGs together. But let's take a look at a typical IgG, and here it is. You have a pair of heavy chains in darker green, so that will be this part right here, 1 and 2. And then you have the light chains in lighter green right here, another pair of light chains. They're connected together. The molecule looks like the letter Y. All antibodies have the same basic structure here. And what's different for each antibody is the tip part. This would be the antigen binding site, and that one is custom made. And that's what takes your immune system some time to figure out how to custom make this antigen binding site, because that one has to be specifically binding, has to bind specifically to the antigen. It has to be custom made. It's like cutting a key, and it does take your immune system some time to figure it out with the clonal selection. So here, now here's our bacterium back with the red circle surface antigen right here, this one, these little circles. And now you have an antibody that has the custom made spot right here for this antigen, the red circle, to fit perfectly into this antigen binding site. And so once that happens, it looks like this. Here we have this antibody right here binding to the red circles on that bacterium. Here you're coating literally these red circles, the bacteria with the red circles with antibodies, and that helps then a macrophage. Once a pathogen has been labeled basically with these antibodies, then the macrophage comes in, and any other phagocytic cell, could be also neutrophil. Then you have what's called enhanced phagocytosis. That means now this phagocytic cell has a really easy time to spot an antibody-coated pathogen, and it will surround it, engulf it, and then internalize it and destroy it. So basically, the coating of the antibody has really helped enhanced the phagocytosis. In addition, the antibodies can also activate antibody-dependent cellular activities. They can activate complement, because once the antibody has bound its target, then the binding site for the first component of complement will be exposed, and then C1 will bind to the stem region of an antibody. And lastly, the antibody can also trigger the mast cell degranulation. That means we're enhancing now inflammatory processes with the release of histamine. So here in steps, so here we have an activated B lymphocyte that has antibodies on its surface, and here it's binding to the red circle on this antigen, this pathogen. And now we're making first plasma cells that are secreting antibodies, and then if we don't need the antibodies anymore, we're just going to keep our memory cells for later use in case we see the same antigen again. It can also act as opsonins, meaning as labeling or tagging antigen for phagocytosis. So here, enhanced phagocytosis, that's definitely a job of antibodies, that they help phagocytic cells to find their targets and surround the targets and destroy the targets, the pathogens. Antibodies, they can also cause an agglutination reaction or clumping reaction. You can see here how several antibodies, they are binding to various pathogens right here and sort of creating this network that ends up precipitating out and agglutinating or clumping. And then here we have that antibodies, they're activating antibody-dependent cellular activity by activating a natural killer cell or an eosinophil. And here on step number 5, the activation of complement. So once an antibody here binds to its target, then the stem portion here on the antibody opens up a binding site for the first component of complement, and then complement will bind and start a complement cascade, and you already know that leads then formation of a membrane attack complex. Also we get enhanced opsonization, and we get enhanced inflammatory processes. And finally here, antibodies, they can also trigger mast cell degranulation, which means that this mast cell right here that's filled up with granules that contain histamine, they release the histamine, and histamine, of course, then also enhances inflammatory processes. Now we're moving on to T lymphocytes. So they are known to carry out cell-mediated immunity or cell-mediated immune functions, and they do this by using contact-dependent signaling. They basically engage into cell-to-cell combat, if you will. They can carry out a variety of different cell-mediated functions, but they tend to engage in sort of cell-to-cell combat fighting. Now T cells, they bind to cells that display foreign antigen fragments as part of the major histocompatibility complex, the MHCs. Now let me say a little bit about the MHCs. We have class 1 and class 2 MHCs. Basically think of them as a self-recognition mechanism, and if you're embedding something into something that you know your immune system will recognize as self, and you're embedding something foreign into it, it helps your immune system to distinguish what it should go after. Now MHC class 1, that MHC complex class 1, we have on all nucleated human cells, and it's recognized by cytotoxic T cells. MHC class 2 is found on macrophages, B lymphocytes, and dendritic cells, and is recognized by helper T cells. So let's take a look at a T cell development, or T lymphocyte development. So we're starting out with the hemocytoblast, this stem cell in the bone marrow, which then eventually leads to a T cell precursor that migrates to the thymus gland if you still have one, if you're young enough to have one still. And then these T cell populations, they just differentiate out into, for example, cytotoxic T cells and helper T cells. So here the example is given: the cytotoxic T cells, they can kill target cells that display the MHC class 1 type histocompatibility complex. And here we have helper T cells. They will latch on to cells that show the MHC class 2 MHC complex. Now, here we have an antigen presenting cell, and this is really this part right here is a very important mechanism, and it shows the central role of helper T cells to the immune response. So basically you have a macrophage right here that has digested, let's say, a bacterium, some pathogen, and it's found the red triangle to be an interesting antigen that was present on that pathogen. And so we had that red triangle earlier, but here it is again. So what the antigen presenting cell will do, it will embed that red triangle antigen from the pathogen. It will embed it into its MHC class 2 complex. And so now here we have the MHC, that's the self-recognition, and then the foreign red triangle on there. And this is basically the target where a helper T cell will latch on. So the helper T cell will then recognize the MHC type 2 complex, and then it will latch on and say, okay, let me check out this red triangle here, it's really interesting, and now I'm gonna go off and make a strategy how we're gonna combat and defeat the red triangle. So this mechanism here shows what happens when you are decreasing helper T cell populations. Think about it. If you are infected with HIV, this virus infects the helper T cells and it will kill them. And now your helper T cell populations are going down, down, down. And now here is your macrophage. It's doing a good job. It's becoming an antigen presenting cell, but there are no helper T cells that will latch on to this antigen presenting cell and figure out a strategy of how to defeat the red triangle. Now, basically, you're eliminating your immune functions. They're still in place, but if there's no helper T cell that activates the other portions of your immune system, for example, to make antibodies or whatnot, then basically you are leaving your forces at home. So it's like being under attack as a country and just going to sleep and saying, well, I don't care, I'm not going to do anything about it until you overrun. So that shows how HIV works and how it causes the acquired immunodeficiency syndrome, because people, they still have the immune functions available, but no helper T cells that are activating the forces that are necessary to fight off a pathogen. Okay, so nice and big here, the summary of T cell activation. So here is our antigen presenting cell. So let's label it APC. And here we are displaying the red triangle. We have our MHC class 2 molecule here, or MHC complex in general, and then there's the red triangle embedded into it. Here comes the T lymphocyte. It latches onto the red triangle, recognizes the MHC as self and the red triangle as foreign, and that interaction then will activate the T lymphocyte. Basically, that interaction of the T lymphocyte with the red triangle right here on this antigen presenting cell, it will set in motion a whole signal transduction cascade that then ultimately activates the entire lymphocyte. So basically, T lymphocytes, they use contact-dependent signaling. As you just saw, this interaction of an APC with a T lymphocyte. And here let's take a look at specifically cytotoxic T cells. They attack and destroy cells that display MHC1 antigen complexes. And again, helper T cells, they really should be renamed to something more important sounding than helper, but they will bind to B cells, promote differentiation into plasma cells and memory cells. They also secrete cytokines that then activate other immune system cells, so they're really central to your